My dear batchmates, my name is Mariam Khader and our seminar topic is PCR. So, what is PCR? PCR stands for Polymerase Chain Reaction. It is a molecular biology technique used to amplify a specific sequence of DNA. Next, we move on to the key milestones in PCR history. In the year 1983, PCR was invented by Gary Mullis. In the year 1985 to 1988, PCR started being used in the labs. In the year 1988, in the year 1993, Gary Mullis was awarded the Nobel Prize for the invention of PCR, and in recent 2020s, PCR was widely used in COVID 19 testing, in genetics, and in crime investigations, and many more. So let's move on to the principle of PCR. Principle of PCR. The polymerase chain reaction principle relies on repeated cycles of heating and cooling to amplify the specific segment of DNA. It involves separating DNA strands, annealing primers to these strands, and extending them with DNA polymerase in order to receive cascade of reactions of DNA amplification. Now comes the components of PCR. There are 6 components of PCR. Number 1, DNA template. It contains the target DNA sequence which want to be amplified. Its source can be a genomic DNA or a plasmid DNA. Next, we move on to the primers. They are short single-stranded DNA sequences of approx 18 to 25 nucleotides that flank the target region. It provides starting points for DNA synthesis. Third, DNA polymerase. A common type of DNA polymerase is tag polymerase, which is thermostable enzyme and obtained from Bacillus thermus aquaticus. Its function is synthesizes new DNA strands by adding nucleotide to primers. Fourth, DNTPs, deoxynucleotide triphosphates. It includes DATP, DTTP, DGTP, and DCTP. Its function is building blocks for new DNA strand synthesis. Fifth, buffer solution, which maintains the optimal pH and salt conditions for the enzyme activity. Last but not the least, magnesium chloride, essential cofactor for DNA polymerase activity. Its concentration must be optimized. Too little or too high can inhibit the PCR. Now I would like to pass down the mic to my other teammate. Thank you.
When we talk about the components of PCR, we should also talk about the steps in PCR. There are mainly 3 major steps in PCR, which are repeated over 30 or 40 cycles. The first step in PCR is denaturation. In denaturation, the double-stranded DNA is heated up to 95 degrees Celsius to make 2 single-stranded DNAs. In the second step of PCR, it is annealing. Annealing, the temperature is lowered up to 50 to 65 degrees Celsius so that the primers can bind to the single-stranded DNAs. Primers are short DNA sequences which target the region to be copied. In the third step of PCR, it is extension. The DNA polymerase, usually the tag polymerase, adds nucleotide to build a new DNA strand starting from the primers. These 3 steps make one cycle, and PCR usually runs for 25 to 35 cycles. After every cycle, the amount of DNA doubles. Types of PCR. There are mainly 4 types of PCR: multiplex PCR, real-time PCR, reverse transcription PCR, and colony PCR. Multiplex PCR is used in detecting multiple pathogens or genes at once. Real-time PCR is used in viral loads, for example, HIV, COVID-19, and gene expression. Reverse transcription PCR is used in the study of the RNA viruses, for example, COVID-19 gene expression. Colony PCR is used in the fast screening of transformed bacteria. Now I would like to pass the mic to my teammate.
Now I talk to you about methods of multiplex PCRs. There are 3 types: primer designs, reactions mixture preparations, and detections of amplified products. In primer designs, multiple primers pair specific for each target DNA regions are designed. Primers should have similar annealing temperatures, no significant complementary with each others. to avoid primer-dimer formations. Reactions mixture preparations. The reactions mixtures contain template DNA, multiple pairs of primerized NTPs, deoxynucleotide triphosphate, tagged DNA polymerase with buffer with MgCl2. Reductions of amplified products. The PCR products are separated using agarose gel electrophoresis. Multiple band of different size indicate applications of different targets simultaneously. Key points of multiplex PCRs allow simultaneous detections of multiple pathogens or genes, save time and reagents, require careful optimizations of primers and conditions to avoid non-specific amplifications. Methods of real-time PCRs. Sample preparations isolate DNA or RNA followed by reverse transcriptions to cDNA if RNA is the targets. Preparations of reactions mixtures contain template DNA or cDNA. Forward and reverse primers DNTPase. Tagged DNA polymerase hot start for better specificity. Fluorescent dye or probe cyber green bind to double-stranded DNA. Tagged man probes buffer with MgCl2.
This is the procedure of reverse transcription polymerase chain reaction procedure PCR. At first, the procedure RNA extraction. The viral RNA was extracted from the sample. Then reverse transcription. RNA was converted to complementary DNA by using the enzyme reverse transcriptase, and then amplification. PCR amplified gene specific to SARS-CoV-2, severe acute respiratory syndrome coronavirus 2. Some examples of this gene are N gene, E gene, RDRP gene, RNA dependent RNA polymerase gene. One point to be noted that they produce RDRP gene because they lack DNA, so they cannot use the host DNA polymerase. And the last, detection. Real-time fluorescence measure the viral load. And now I pass the mic to my next partner.
Next step is the colony PCR. First step is selection of colonies, in which a single bacterial colony is picked from agar plate using a sterile toothpick or paper tip. Next step is the cell lysis, which has two methods. First one is the direct method. Touch the colony into 10 to 20 microliter sterile water or lysis buffer in a PCR tube. Then heat at 95 degrees Celsius to 5 to 10 minutes to lyse the cells and release the DNA. Spin briefly to pellet debris. Use supernatant as PCR templating. Next is the alternative method. Directly dip the colony into prepared PCR mix and proceed, but only for robust tag polymerase. Next is the application of PCR. First is the medicine field, used for testing of genetic disease mutations, monitoring the gene in gene therapy, and detecting disease-causing gene in parents. Second is forensic science, used as tool in genetic fingerprinting, identifying criminals from millions of people, and paternity tests. Last one is research and genetics, to compare the genome of two organisms in genomic studies, analysis of gene expression, and gene mapping.
Suppose during COVID time, you said that real-time PCR is used in COVID time. So there, we used to see the result, like the viral load 22, 32, 36, 14, 12. What does it mean?
If the viral load is beyond the threshold, that signifies that it is a positive result of COVID.
So suppose you and your friend both have got COVID results. Suppose your value is 12 and your friend's value is 24. So who is having the disease and who is having less disease? Yeah, what is the requirement for this tagged DNA polymerase?
Sir,
We have so many DNA polymerases.
Sir, it's a thermostable enzyme which can maintain the separation of DNA strand. So that is why it is used, because it is thermostable.
The tagged DNA polymerase is isolated from some special bacteria. Where is it found?
Bacillus thermocycloticus.
Where is it found? What is the source?
Sir, it is found in seaweeds. So,
Hot spring.
hot springs.
Hot spring. Why? See, there is a story behind how it was invented, discovered by Karimuli. So he went to a vacation where he found that hot spring, and there he isolated the bacteria from where he got the thermocycloticus. So here, later on, it was found in many bacteria, but initially it was discovered from this. Anyway, presentation was good. You all are prepared good. But thing is that value should be less is more harmful.
Yes, here the technique of amplification. Okay. So how many copies will be produced by 20 cycles? And
This also I explained yesterday.
how many cycles theoretically? How many number of copies are possible after 20 cycles?
I told you yesterday to calculate in the evening, in the afternoon, just to make a calculation. And
After 20 cycles, it will produce 1 million copies. That is a lakh. And after 30 cycles, it will produce 1 billion copies. But it is theoretical. Practically, so many copies are not produced because of many factors.
অত reagent কে দেবে? Who will give those much of reagent?
How many? What are the characteristics of the primers here? What is primer? What is primer?
Sir, it helps in
You say PCR, okay? Primer is necessary for PCR. And what is the characteristics of the primer?
You have said in the presentation.
primers are short single-stranded DNA structures of approx 18 to 25 nucleotides, which helps in adding the
What is the necessity of the primer? Yes, you say the criteria of the primer you fulfill. It should be complementary to the franking sequence of the target DNA. আসলে primer হবে না তো ফ্র্যাঙ্কিং সিকোয়েন্সে কমপ্লিমেন্টারি হবে তাহলে তবেই তো ওটা অ্যাটাচ করবে এবং তারপরেই তো ওটা এলোঙ্গেট করবে
Now, sir said about primer which recognizes the flanking region of a DNA sequence, the target DNA sequence. But how do you get sure about that it is synthesizing the correct sequence? Is there any other method which can be employed during PCR? You need to isolate a sequence and you need to be sure that it is correct. No mutation, nothing, no mistake is there. Because in this process, that DNA polymerase, there is no question of proofreading. So they will just go on writing what is written. But how do you make sure that the sequence you have processed with PCR is there? How do you understand?
তোমার ঠিক প্রাইমার সিলেকশন হয়েছে কিনা কি করে বুঝবে
So there is a point called internal primer. There is a point called internal primer. To double check the sequence is correct or not, we always use an internal primer. In between two flanking primer, a small internal primer is also used. That makes sure that the target sequence is always correct. It is not mistakenly replicated or copied. Okay. Anyway.
What is the biggest disadvantage of the PCR? যেটা নিয়ে আমরা খুব ভাবি biggest disadvantage.
Disadvantage of PCR. All advantages you say.
ওটার জন্য আমরা PCR এ এত প্রোডাক্ট আমরা পাই না টার্গেট ডিএনএ যতটা ডিজায়ারেবল হওয়ার কথা
sir, it
কত
easily gets
নাম্বার অফ কপিজ
contaminated.
কেন পাই না আমরা
বলুন।
মানে এফিসিয়েন্সি অফ দ্য পিসিআর যতটা আমরা থিওরিটিক্যালি ভাবছি প্র্যাকটিক্যালি কিন্তু আমরা সেরকম দেখি না তার কারণ হচ্ছে কি ওয়ান অফ দ্য বিগেস্ট চ্যালেঞ্জ হচ্ছে কি সেটা হচ্ছে কন্টামিনেশন টার্গেট ডিএনএ এর সঙ্গে যদি অন্য ডিএনএ যদি কন্টামিনেট হয়ে যায় হ্যাঁ সেক্ষেত্রে কিন্তু আমি বেশি পাবো না
সেখানে ইন্টারনাল প্রাইমার
ইন্টারনাল প্রাইমার ইউজ করা হবে আবার প্রাইমার যদি দুটো প্রাইমার একটা সেট অফ প্রাইমার আমরা ইউজ করছি দুটো প্রাইমার, ঠিক আছে? এবার দুটো প্রাইমারের মধ্যে যদি হোমোলজি থাকে তাহলে প্রাইমার ডাইমার ভালোসে না, তাহলে এটাও দেখতে হবে দুটো প্রাইমার যাতে দুটো প্রাইমারের মধ্যে যদি কোনো কমপ্লিমেন্টারিটি না হয়, ঠিক আছে? দুটো প্রাইমার যদি কমপ্লিমেন্টারি হয়ে যায়, কিছুটা কমপ্লিমেন্টারি হয়, দুটোটা না হলো, তাহলে গেলে প্রাইমারের সঙ্গে প্রাইমারের অ্যাটাচমেন্ট হয়ে যাবে, যে ডাইমার তৈরি করবে।
So it will continue copying the primer only.
হ্যাঁ, হবে না multiplication। অনেক ডিসঅ্যাডভান্টেজ আছে। আমরা ওই যে বললাম 20 সাইকেলে 1 মিলিয়ন 30 সাইকেল,
Suppose you have gone to market to purchase an PCR machine. What will you call for it? Ask for it.
মেশিনটা কি বলে?
What is the name of the instrument?
মানে এটা সাইক্লিক্যালি টেম্পারেচার চেঞ্জ করা হয়।
Sir is giving hint.
সাইক্লিক্যালি টেম্পারেচার চেঞ্জ করা হয় 95 টু 55 টু 72। এই যে সাইক্লিক্যালি টেম্পারেচার চেঞ্জ করছে একটা অটোমেটেড মেশিন দিয়ে করা যায়। হোয়াট ইজ দ্য নেম অফ দ্য মেশিন?
Thermocycler.
Thermocycler. Very good. Very good. Hey, others should also participate. They are not anyway. Anyway, you can go. Thank you. Next.
Thank you, sir.
A very good morning, everyone, to all my fellow batchmates and my respected teachers. Today we'll be presenting on the topic prokaryotic gene regulation. So let's hop on to that. Imagine if you had to wear every single piece of clothing you own every single day, in spite of the circumstances, in spite of the occasions. This sounds very absurd, right? So what would have happened? That what would have happened for a bacterial cell to express every single gene it has. So that's when the prokaryotic gene regulation comes to play. Introduction to the gene regulation in prokaryotes. As we know, in prokaryotes, gene regulation is very vital for adapting to changing environmental conditions. Unlike eukaryotes, prokaryotes such as E. coli, they lack nuclear membrane that allows tight coupling between transcription and translation. This makes regulation at the transcription level very efficient. Prokaryotic cells have very limited resources, so they manage their limited resources very wisely. Expressing all the genes at all the time would be very energetically expensive and unnecessary. Thus, they conserve energy and materials and proteins that are synthesized only when required, and also the cell that can adapt quickly to environmental changes such as nutrient availability. This regulatory ability is very crucial for survival and competitive advantage in very diverse conditions. Then we'll come to the mechanisms and levels of gene regulation. So gene expression in prokaryotes is primarily controlled at the transcription initiation stage, though other levels also do exist, namely transcription control, post-transcription control, translational control, and post-translational control. At the transcription control is the most efficient and critical level of control in bacteria. This determines whether a gene will be transcribed into mRNA at all or not. The post-transcription control includes the control of RNA degradation and the processing of it. The translational control regulates the initiation of the protein synthesis. The post-translational control involves the initiation, activation, and degradation of the proteins after they're made. Among this, the transcription regulation is the most studied due to its direct control over the flow of genetic information from the DNA to the RNA. Thank you. I'll pass the mic to my teammate.
We will be discuss about the Operon Model of gene regulation. Lac operon means the array of gene involved in metabolic pathway in lactose in E. coli. The cluster of gene, lac operon is described by Jacob and Menoth in 1961. This cluster of genes transcribed from a single polycystic mRNA. This can be translated into many polypeptide. The component of operon, structural gene, codes for functional enzyme, promoter, binding site for RNA polymerase, operator, binding site of repressor protein, and regulatory gene or LAC-I gene, codes for repressor protein. Types of operon: inducible operon, usually of turn on presence of substrate; repressible operon, usually on turn on presence of product accumulation. Structural and functional of the lac operon. Structure and function of the lac operon. Lac operon in E. coli, the classic example of inducible operon used for metabolism of lactose gene in the lac operon, lac Z gene, lac Y gene, lac A gene. Lac Z gene codes for beta galactosidase used hydrolyze lactose into glucose plus galactose. Lac I codes for protease to facilitate lactose entry into the cell. Lac A codes for transacetylase which detoxify by product of lactose metabolism. Regulatory element lac I gene, a separate gene code for repressor protein that bind to operator and block transcription. Promoter site for RNA polymerase bind and initiate transcription. Operator DNA region where the repressor bind and inhibit transcription. In the absence of lactose, the repressor bind to operator, block the transcription. In the presence of lactose, the operon is induced, allow the enzyme production.
Any diagrams? You have not. You have to explain diagrammatically. That is position of the lac Z, lac Y, lac A, lac I, promoter, operator. Have you not Vincent? Vincent, you did.
Hey, what is that? One question.
The diagram to get the picture to one diagram mentioning all the features. You have mentioned in the yes, that diagram. Do বড় করো বড় না করা যাবে না
See, the thing is, the diagram should have been bigger than the letters.
তোমার এই কথাগুলো থেকে ডায়াগ্রামটা খুব পরিষ্কার হওয়া যায় ঠিক আছে কথাগুলো তো তুমি বুঝতে বলছো ডায়াগ্রামটা একটু ভালো করবে
So this is regulatory structural gene, lac Z gene, lac Y gene, lac A gene.
ঠিক আছে
ঠিক আছে
আমি
ডায়াগ্রামটা উঠে দেখি
বুঝে গেছে ছোট করে দিচ্ছে।
বলো প্রত্যেকটা সংখ্যা বলো হোয়াট ইজ বিটা গ্যালাক্টোসাইডেস কি করে
Sir, lactose के ग्लूकोज प्लस
কি করবে
lactose के ग्लूकोज प्लस ग्लैक्टोज कन्वर्ट করবে
yes you convert the glucose lactose into glucose and
গ্ল্যাকটোজ
glucose and
গ্ল্যাকটোজ গ্ল্যাকটোজ
lactose ঠিক আছে আর what is the function of transacetylase
ट्रांसएसिटाइलेज सर बाय प्रोडक्ट ऑफ लैक्टोज मेटाबॉलिज्म के डिटॉक्सिफाई করবে डिटॉक्सिफाई করবে बाय प्रोडक्ट ऑफ लैक्टोज मेटाबॉलिज्म
The lac operon regulates under three modes. When only glucose is present. Second, when both glucose and lactose are present, and third, when only lactose is present. When only glucose is present, the lac I gene, which produces the repressor proteins, it continuously produces the repressor proteins, which gets binded to the promoter gene and inhibits the further on translation because the RNA polymerase does not get to bind with the operator region anymore. And when both the lactose and glucose are present, the presence of glucose inhibits the production of adenine cyclase, which reduces the CAMP levels. And due to the short levels of CAMP, there is no production of the CAMP proteins
which are the catabolite activator proteins. It does not bind with the cap, and no complex is formed. The cap-cam complex, it is not formed anymore. So the RNA polymerase does not, the RNA polymerase is not binded with the operated gene anymore and is replaced.
Lack operon regulation when both glucose and lactose are present. Lack operon induction when only lactose is present. When glucose is absent and only lactose is available, the lack operon is fully activated. The steps are: lactose or its isomer allolactose binds to the repressor protein, causing a conformational change. The repressor detaches from the operator, allowing RNA polymerase to access the promoter. Lack of glucose increases CAMP, which binds to CAP. The CAP-CAMP complex binds near the promoter, further enhancing RNA polymerase binding. Transcription of LAC-Z, LAC-Y, and LAC-A occurs. The resulting enzyme is metabolized lactose. Beta-galactosidase helps lactose to convert glucose. Lactose converts to glucose plus galactose. Permease helps in lactose entry. Trans-acetylase helps in detoxification rule. This is a coordinated example of negative inducible and positive control working together. Summary and importance of lack operon regulation. The lack operon is central model in molecular biology that illustrates the elegant control mechanisms in prokaryotic gene regulation. The key points are: the lack operon is an inducible operon regulated both negatively by repressor and positively by CAP and CAMP. It ensures the enzymes are produced only when lactose is present and glucose is absent. This avoids unnecessary expenditure of cellular energy. The lack operon demonstrates principles such as feedback inhibition, catabolite repression, gene-environment interaction. The applications are used in molecular cloning, example IPTG as lactose analog inducer, foundation of understanding operon-based gene regulation in biotechnology and synthetic biology. Thank you.
Complete. First of all, this is incomplete. There is another model called attenuation. You have missed. Second of all, the PowerPoint presentation was horrible. This is the one of the poorest way of presentation.
So this wasn't the actual thing. The one we made.
Doesn't matter. What you have shown, that is final.
So after editing it,
You can't say I had Amitabh Bachchan at house, but I just presented Abhishek Bachchan. No. Do you understand what you have spoken about? So, what is polycystic mRNA?
বাংলা থেকে বলবো
বলো।
একটা এমআরএনএ যখন একাধিক মানে সিস্টোন থাকে, মানে অনেকগুলো মানে মোর দেন ওয়ান সিস্টোন থাকে তো কোড করে, মানে কোড মোর দেন ওয়ানকে কোড করে, মানে মোর দেন ওয়ান পলিপেপটাইডকে কোড করে, পলিপেপটাইড অর প্রোটিন একটাকে কোড করে। হ্যাঁ, ওয়ান পলিসিস্টোনিক মধ্যে মোর দেন ওয়ান প্রোটিনকে কোড করবে স্যার। হ্যাঁ, ওটা মনোসিস্টোনিক। পলিসিস্টোনিক।
প্রোক্যারিওটিক রেগুলেশনের একটা মডেল আছে যে বললে তুমি, কিন্তু ইউক্যারিওটিক রেগুলেশনে মানে আমাদের কোনো মডেল কিন্তু নেই। কেন বলো তো? কিরকম মনে হয়, কেন আমাদের মডেল হলো না? কেন আমরা ব্যাকটেরিয়াল মডেল জেনে কি করব? আমাদের মডেল কোথায়? আমাদের মডেল বার করাটা খুবই কঠিন কাজ, কারণ ইউক্যারিওটিক রেগুলেশন ইজ মেনি মাচ মোর কমপ্লেক্স than prokaryotic one. এত সোজাভাবে ইউক্যারিওটিক মডেল তৈরি করা যাবে না।
So what is the basic difference in transcription translation in prokaryote and eukaryote?
স্যার প্রোক্যারিওটের ক্ষেত্রে ট্রান্সক্রিপশন আর ট্রান্সলেশন স্যার সাইটোপ্লাজমে হয়, আর ইউক্যারিওটের ক্ষেত্রে ট্রান্সক্রিপশন নিউক্লিয়াসে হয় আর ট্রান্সক্রিপশন স্যার
আচ্ছা নিউক্লিয়াসে হয় তারপর
আর ট্রান্সলেশন হয়।
নিউক্লিয়াসে ট্রান্সক্রিপশনের পরেই কিন্তু ট্রান্সলেশন হয় না।
হ্যাঁ
they are separated by
এর ক্ষেত্রে কাপেলড হয়
space and time.
হ্যাঁ প্রোক্যারিওটিক এর ক্ষেত্রে কাপেলড হয় ট্রান্সক্রিপশন এন্ড ট্রান্সলেশন কাপেলড হয় মানে সাইমালটেনিয়াসলি হয়
Coupled আর এখানে ডিফারেন্ট
কাপল এগুলো বলা কাপল মানে কি
সাইমালটেনিয়াস একসাথে হয়
ট্রান্সলেশন একসঙ্গে ঘটবে প্রায় একসঙ্গে ঘটবে, কিন্তু এখানে বিপরীতে তা হবে না।
that is why it is very easy to regulate prokaryotic gene, but that is why it is very difficult to regulate eukaryotic gene.
বিপরীতে অনেক ফ্যাক্টর কাজ করে, অনেক ফ্যাক্টর।
Okay, anyway, so next time you just keep in mind this is not the way you prepare slides.
বলতো কত রকম মানে জিন এক্সপ্রেশন আছে। একটা যে কনস্টিটিউটিভ, how do you mean by constitutive expression?
কনস্টিটিউটিভ
হ্যাঁ
স্যার কনস্টিটিউটিভ এক্সপ্রেশন হচ্ছে স্যার ল্যাক আই জিনটা করে
হ্যাঁ
কনস্টিটিউটিভ ল্যাক আই করে মানে সব সময় যে স্যার মানে এক্সপ্রেস হতেই থাকে স্যার
এক্সপ্রেস কনস্ট্যান্টলি উইদাউট এনি রেগুলেশন।
No regulation.
সব সময় এক্সপ্রেস যেরকম ল্যাক আই জিনটা থাকে
এরকম কোনো আছে আমাদের বডিতে এরকম কোনো জিন আছে যেটা কনস্টিটিউটিভলি এক্সপ্রেস হয়, কোনো রেগুলেশন ছাড়া এক্সপ্রেস হয়, এরকম কোনো এই এনজাইম আমাদের বডিতে আছে?
হেক্সোকাইনেজ
আরে যেগুলো আমাদের প্রতিনিয়ত হচ্ছে যেমন ক্রেব সাইকেল,
হ্যাঁ
গ্লাইকোলিসিস, হ্যাঁ এগুলোর কিন্তু রেগুলেশনের জন্য কোনো মেকানিজম নেই, এগুলো কনস্ট্যান্টলি এক্সপ্রেস হয়ে যাচ্ছে as per বডিজ নিড। আর একটা হচ্ছে গিয়ে রেগুলেটেড এক্সপ্রেশন, তুমি যেটা বললে সেটা হচ্ছে রেগুলেটেড। টিস্যু স্পেসিফিক এক্সপ্রেশন, টিস্যু স্পেসিফিক এক্সপ্রেশন মানেটা কি?
স্পেসিফিক এক্সপ্রেশন
টিস্যু স্পেসিফিক এক্সপ্রেশন অফ জিন
কিছু নির্দিষ্ট সাবস্টেন্সের জন্য এক্সপ্রেস করবে।
আমি একটা উদাহরণ দিচ্ছি তাহলে বুঝতে পারবে। ইনসুলিনের জিন সব জায়গাতে আছে, সব সেলে আছে, ঠিক আছে, কিন্তু এটা এক্সপ্রেস হয় একমাত্র প্যানক্রিয়াটিক দুটা সেলে। এইটাকে বলে টিস্যু স্পেসিফিক এক্সপ্রেশন। অ্যাকচুয়ালি সমস্ত জিনই কিন্তু বেশিরভাগ জিন, মোস্ট অফ দ্য জিনস আর এক্সপ্রেসড in the tissues selectively. সব সেলে কিন্তু সব জিন এক্সপ্রেস হয় না, পার্টিকুলার কিছু সেলে ওই জিনটা এক্সপ্রেশন হবে, যেমন ইনসুলিন জিন দুটা সেলে এক্সপ্রেস হবে। এগুলো হয় মেলানিন স্কিনে হবে, থাইরয়েড হবে, টিএসএইচ এক্সপ্রেশন থাইরয়েড সেলে হবে, টিএসএইচ টা থাইরয়েড সেলে হবে না। টিএসএইচ কোথায় তৈরি হয় বলো তো?
টি
থেকে তৈরি হয়, টিএসএইচ কোথা থেকে আসে বলো তো?
TSH কোথা থেকে আসে? TSH এর সোর্স কোথায়?
স্যার পিটুইটারি
TSH
পিটুইটারি থেকে
থাইরয়েড স্টিমুলেটিং হরমোন কোথায় তৈরি হয়?
পিটুইটারি
পিটুইটারি। Adenohypophysis and neurohypophysis?
অ্যাডিনোহাইপোফিস
অ্যাডিনো, হ্যাঁ ঠিক আছে, নাম এবার নাম হচ্ছে নাম পরজন নেক্সট আয়
Good morning, respected professors and one and all present over here. I'm Nancy Neeraj Patel, and let us proceed with the topic of cardiac marker in today's session. So, what exactly is cardiac marker? Cardiac markers are biochemical substances, say enzymes or protein or hormones, which get leaked in the circulation whenever any injury is happen to the cardiac muscles. The definition goes like cardiac markers are biochemical substances released into the bloodstream when the myocardium is injured. They are used in diagnosis, monitoring, and management of the conditions like myocardial infarction and other cardiac dysfunctions or diseases. Later on, we have uses. What are the uses of cardiac markers? Cardiac markers are importantly used to detect the myocardial infarction. Further on, they also help us to detect the myocardial reinfarction that is about to happen. It helps us predict the future possibilities of myocardial infarction as well. It helps in guiding treatment decision. It helps us to monitor the progress in the treatment. Then it helps us to distinguish cardiac from non-cardiac diseases. Chest pains. Also, it helps to estimate the severity of the damage occurred. Later on, what makes a good cardiac marker, or what are the characteristics of an ideal cardiac marker? First of all, it needs to be highly specific. It should only detect cardiac muscle injuries and should not elevate whenever the other injuries are happening, other body or muscle injuries are occurring. Then later on, it should be also be sensitive. For example, troponin. Troponin is highly cardiac muscle sensitive cardiac marker. Later on, it should rise early after the injury. It should detect even the smaller injury in the cardiac muscles. For example, myoglobin. Myoglobin detects even the minor injuries and rise within the span of 1 to 2 hours. Then it should peak and stay elevated for enough time so as the doctors could predict when the cardiac injury or myocardial infarction exactly occurred. Later on, it should be easy to detect in blood. Also, it should stay stable in blood, so as the doctors could diagnose diagnose the cardiac muscle injury, even when the tests are done late. It should be proportional to the extent of heart damage. It means that as much higher are the level of the cardiac marker found in the circulation, that much severe is the injury. Later on, we have this table for cardiac markers, major cardiac markers. First of all, we have troponin I and T. It rises it rises after the span of 3 to 6 hours and reaches its peak within 12 to 24 hours. Later on, it goes back to normal within 7 to 14 days. Later, we have CKMB, which rises in 3 to 12 hours, reaches its peak in 12 to 24 hours, and goes back to normal in 2 to 3 days. Myoglobin rises within 1 to 2 hours, which is pretty early. Then it peaks in 4 to 6 hours and goes back to normal in 20 to 24 hours. Then we have LDH, which rises in the span of 12 to 24 hours, reaches its peak in 24 to 36 hours, and goes back to normal in 10 to 15 days. And then we have AST. AST rises in the span of 6 to 12 hours, reaches its peak in 24 to 36 hours, and goes back to normal in 4 to 6 days. Digging up more into the mechanism of how cardiac markers are releases in the blood, whenever cardiac muscle gets injured, as in case of myocardial infarction, the cardiac muscles are compromised cause of say ischemia of heart or cardiac muscles or necrosis, or the hypoxia. The cardiac muscle gets damaged and the cytoplasm ruptures. Sarcolemma of the cardiac muscle ruptures, and all the fluid which is inside starts getting into the circulation, and that's how cardiac markers are diagnosed in the blood. Then we have troponin. Troponin is an ideal cardiac marker and is considered as golden standard for diagnosing heart attack, as it is a most specific and sensitive marker and is only found in cardiac muscle cells. It remains elevated up to 2 weeks, and it is also observed to be mildly elevated in kidney diseases and heart failures. Then we have CKMB. CKMB is found in cardiac muscles, as well as a small amount of it is also observed in skeletal muscles. It also helps in diagnosing reinfarction, as all the other cardiac markers go back to normal all the cardiac markers stay elevated for pretty long period of time. Meanwhile, CKMB, that is creatine kinase MB, stays elevates early, go back to normal range, and then starts elevating again whenever the reinfection is about to happen. So that's how it helps in detecting cardiac reinfarction, that is second heart attack. Then it is less specific, as it also may rise in skeletal muscle injuries, as small amount of it is also present in skeletal muscles. Then we have myoglobin. Myoglobin is also known as early marker. It is known as early marker as it rises within 1 to 2 hours of cardiac injury, and it is also found in skeletal muscles. So evidently it is not, as it is sensitive, but it is not specific, as it may also rise in case of muscle injury, and alone it is very rarely used alone during diagnosis. Then we have LDH1, that is lactate dehydrogenase. It is present in many tissues like heart, liver, kidneys, RBCs, and other muscles. The older marker rises, it is an older marker and it rises late. It follows a flip pattern, that is when when the ratio of LDH1 is higher in the blood as compared to LDH2, it indicates myocardial infarction. It is now very rarely used, as there are better markers like troponin present in are available. Then we have AST, aspartate transaminase. It is also present in many tissues like heart, kidney, liver, brain, and skeletal muscles. It is also not specific, as it also rises in liver diseases and other muscle injuries. It also has been replaced by several other new markers available. Then we also have new emerging markers: heart type fatty acid binding proteins, glycogen phosphorylase BB, and copeptine. These are not being used routinely, but they do show promises. Then again we have clinical applications, that is first of all cardiac markers are used in diagnosis of myocardial infarction. Later on they also help in diagnosing or detecting reinfarction which is about to occur. First of all, doctors go for ECG as immediate test for myocardial infarction, and then they do check the troponin levels. If they are elevated, it confirms a myocardial infarction. Then we have risk stratification in acute coronary disease, acute coronary syndrome, and it also helps in differentiating the causes of myocardial injury. How to approach a chest pain. If someone is going through a chest pain, how do doctors approach them? First of all, they go for taking ECG, which is an immediate test. After observing the results of ECG, they go for observing ST segment. If the ST segment is elevated, they confirm it as STEMI, that is ST segment elevated myocardial infarction, in which it occurs whenever the major coronary artery is completely blocked due to atherosclerosis, collection all the fatty when it gets accumulated and the lumen of the artery becomes narrow. And if ST segment is not elevated, they go for checking troponin levels. If the troponin levels are elevated, it confirms not elevated ST segment,
non-ST elevated
ST
MI.
segment elevated myocardial infarction. And if everything is normal, the test is repeated after 6 hours, and otherwise it is ruled out. In all, we have a short summary of all the cardiac markers that we have observed till now. First of all, we have troponin. It is evidently considered as best cardiac marker, as is really sensitive and specific. Then we have CKMB, which helps us in detecting myocardial reinfarction. Then we have myoglobin. It rises early, but it is not specific. And LDH and AST are outdated. Thank you.
Good.
Very good coverage.
Sir,
You alone have covered almost everything.
yes.
Very good coverage. Many information you have given to us. Now let us find a problem. The patient comes with chest pain 1 hour after chest pain. 1 hour after onset of the chest pain comes to the emergency. On ECG, you find still elevated. Which marker will be elevated in this case?
Within-
Raw blood cell.
Within 1 to 2 hours, myoglobin is seen to be raised.
Myoglobin will be
Is elevated. Is
myoglobin
observed to
will
be.
be elevated, not others.
Not others. Others like troponin.
If patient with chest pain comes 1 hour after he draw the blood for myoglobin, it is negative. What will be your inference?
I'm sorry, sir.
Chest pain means
Sir,
patient comes with chest pain.
yes, sir.
Patient comes with chest pain.
Yes, sir.
You estimate myoglobin.
Yes, sir.
Myoglobin is
First of
negative. What will be your inference?
So patient, we would reconduct the test after 6 hours.
It is not myocardial infarction, number 1.
It is not.
Because myoglobin is very much sensitive, but not specific. If myoglobin is not present, myoglobin is usually present very little amount, negligibly, by almost 0. When it is elevated, it indicates some injury, either cardiac muscle or skeletal muscle. Even a single click of injection will
increase myoglobin.
will get elevated. So it is not specific. But the negative myoglobin is very essential to exclude myocardial infarction. Negative myoglobin.
In one point you have shown CKMB. You go to the slide.
Now CKMB, how to understand?
Yeah, here. What is this? It is, you said, reinfection. The test done for reinfection. So in CKMB, what do you measure?
What do we measure?
The total CKMB or what? What is the name of the parameter?
What is
Which is the name of the parameter which is used for understanding whether it is reinfection or not.
So we go for
Because CKMB is already elevated. Okay?
it was already
And it might go down also from normal level, from elevated level. So how do you know that it is after elevation or after first elevation or after second elevation? How do you know?
we confirm it after the second elevation conducted after few days.
And,
2 to 3
there
days.
is a special parameter about CKMB. It is the total CKMB which is inactive, not active. The inactive protein also is measured. How much total CKMB has been released by the tissue. That gives a better picture of how much muscle is infarct in the heart. Okay?
Yes.
It is not just enzymatic activity is measured. CPK has. CPK
CPKMB. How does it differ?
What, sir?
Total CPK and CPKMB. How does it differ?
Total.
नीचे नाम नीचे.
What do you mean by CPK? Total CPK. What do you mean by total CPK? Total CPK is a function of 3 enzymes, isoenzymes. What are those?
MM, MB, and BB.
MM, MB,
And BB.
and
BB.
DB.
Yes.
Yes.
Brain.
Then total CPKMB, total CPK rises.
Yes.
Is it possible for the case of myocardial infarction
No, sir.
to increase CPKMB without increase of CPK, total CPK? Is it possible? Total CPK is normal, but CPKMB is elevated. Is it possible?
Is it possible or not?
No.